as most of you will know, PD Frontline is an online Parkinson's study, and our main purpose is to be a platform for people with Parkinson's to become essentially trial ready. So we want to recruit as many people as possible who want to take part in research and who wish to know whether or not they carry a genetic risk factor for Parkinson's, to then inform them of relevant trials and studies they can participate in. So there are three main purposes of PD Frontline: to genotype, to act as a registry, and then refer on to clinical trials and studies. So we want to slow down the progression of Parkinson's disease, and the way that trials are moving, there's more focus on gene-targeted therapeutics. The two genes that we test for are the GBA and LRRK2 genes. GBA is the most common risk factor gene for Parkinson's, but most people are unaware of whether or not they carry the change in the GBA gene. So we want to develop a cohort of genotyped individuals who can then be referred on to future trials and also clinical observational studies as well. So this is quite important because, as I mentioned, PD research is at a very exciting juncture where for the first time the possibility of drugs that could stop or slow down the progression of Parkinson's is a real possibility. So we want to be able to build a cohort of as many genotyped individuals as possible so that when clinical trials or studies become available, we have a whole cohort ready to take part. So I mentioned the GBA gene, but some of you may not know what that is. So I'm going to briefly go over what the GBA gene is, how we inherit the GBA gene, the change in the GBA gene, and what do we know about the relationship between the GBA gene and Parkinson's so far. So we each inherit 2 copies of every gene, one from each parent. This is the same with the GBA gene. People with Gaucher's disease, however, have a genetic alteration in both copies of the GBA gene, and they will pass this on to their child. But someone with Gaucher's will have a genetic alteration in both copies of the GBA gene, so if each parent carries a copy, they would have a 50% chance of developing Gaucher's. People with a genetic alteration in one copy of the GBA gene will not develop Gaucher's disease. This is known as a carrier. They have a slightly increased risk of developing Parkinson's, but this is still a very low risk, and most people will not develop Parkinson's disease. So as you can see here, one gene from each parent means that you have a 50% chance of carrying the GBA gene. So what do we know about GBA and Parkinson's so far? So first of all, the GBA gene makes the enzyme glucose cerebrosidase, or GCase. A lack of this results in a harmful buildup of certain fats and nutrients throughout the body. Alpha-synuclein is a protein that builds up in Parkinson's and causes nerve and cell damage. So what we know so far is that when there's a decrease of GCase, there's an increase of alpha-synuclein. So where there is not a change in the GBA gene, where there is a normal working functioning GBA gene, the alpha-synuclein is broken down and we don't see this nerve and cell damage. But when there is a fault in the GBA gene, the alpha-synuclein is not broken down. Instead, it builds up in the cell and it clumps together, resulting in this nerve and cell damage. So what does the study involve? So we currently have over 2,300 participants, and we recruit all across the UK. Our study continues to grow very rapidly, and for that, I thank you very much for continuing to participate in PD Frontline. Who can take part? Those diagnosed with Parkinson's disease, those who live in the UK, and do not know if they carry the change in the GBA gene. If you are aware that you carry the change in the GBA gene, you will be eligible for our sister study, Rhapsody, which I know some of you may belong to. Signing up to PD Frontline is very quick and simple, taking just 10 minutes. It requires registration, filling out a consent form, and completing a very short survey. The next steps involve us sending you a saliva collection kit, which you will then complete and return to us in a prepaid envelope. The sample is then sent off for genotyping and analysis, and then we do inform you of your results once they become available. So I just wanted to provide a brief overview of PD Frontline and the next steps once you have your results and what this can mean for you. So As I mentioned, PD Frontline will act as a platform, referring individuals to studies where they may be eligible for. So, for instance, those of you who may be GBA positive will be referred on to the RAPSI study, which is a longitudinal observational study collecting data yearly. This is all done online and involves clinical assessments and a biannual smell test. This will collect data about the progression of your Parkinson's, both with and without the GBA gene, to see how the GBA gene may be affecting Parkinson's symptoms. All participants of both studies will also be referred to appropriate clinical trials and observational studies. For instance, as part of PD Frontline and Rhapsody, we have had several sub-studies. For instance, this year we had the PD Biome study, which looked at the interaction between the gut and microbiome, and all their participants were recruited through Rhapsody and PD Frontline. This was purely an observational study and not a clinical trial, but it's just an example of different ways you may be involved. And these are just the contact details at the end if you want to get in touch if you have any questions. Okay, so thank you all for listening. Next on the agenda is Professor Schapira. Professor Schapira is the principal investigator of both the PD and Frontline and Rhapsody studies. He is head of Department of Clinical Movement Neurosciences at the Institute of Neurology. He will be discussing the upcoming phase 3 of the Ambroxol trial.
Thank you very much indeed, Selen. So I've just got a couple of slides, and I just wanted to emphasize something that Selen said in her excellent introduction to this area, which is that research in Parkinson's disease is changing dramatically. And this is because we have been able to identify genes which are responsible for increased risk in developing the disease. And most important amongst all of these is the GBA gene. We find the GBA gene in about 12% of Parkinson people in the UK, and it's probably a similar figure around the world. So it's by far the commonest. The next commonest, or probably around 1%, is the LRRK2 gene, which we also test for. Now, the change in Parkinson's research that's been so dramatic and is accelerating our work towards developing drugs to slow the disease is based upon these gene identifications, so the GBA and the LRRK2, and new treatments are already now being tested for both these disorders. So what's really interesting is that designing a drug, for instance, for those with GBA may very well have benefits for even those people with Parkinson's without a GBA gene. And the reason for this is because the drugs for GBA-related Parkinson's disease seem to also have benefit on the same pathways in patients and people without GBA mutations. Of course, we can do an awful lot of work, as we have done in the laboratory and using all sorts of different models, but ultimately we need to know whether what we're developing is safe and beneficial for patients, and that's why we need your help and why we set up PD Frontline. And as Selen has said, the moniker is to get trial ready. And I'm going to focus just for a few minutes on a drug called Ambroxol. And Ambroxol is a very interesting compound. It was found almost by chance to be beneficial in reversing the effects of mutations of the GBA gene. So mutation of the GBA gene, an error in the gene, will result in certain changes in the cell. And Ambroxol was found to be capable of reversing those and restoring the function of the cell to normal. In particular, in the context of Parkinson's disease, it was found that Ambroxol was capable of reducing the alpha-synuclein levels in the cells in the brain and Selen reminded you that this protein alpha-synuclein is important in Parkinson's disease because it's this protein that accumulates in the brain and is thought to cause nerve damage. So if I could have the next slide, please. What we did a couple of years ago and published in 2020 was what we call a proof of principle trial of Ambroxol. Let me tell you a little bit about Ambroxol itself, first of all. This drug has been around for about 40, 50 years. And it was, first of all, produced as a drug for respiratory problems, in particular for cough. And so over time, it's been found to be very simple, effective, and importantly, a very safe drug. So much so that in Europe, for instance, you can buy it over the counter. You don't need a prescription for Ambroxol. So it's got the advantage of being used for many years and being very safe. What we did in this study with a relatively small number of people with Parkinson's disease, both with and without GBA mutations, was to see if the Ambroxol was well tolerated in this patient population, safe, whether it got into the brain, because of course we need the drug in the brain as opposed to in the lungs, where it's previously been found to be effective, and to see what effect it had on alpha-synuclein. And I'm pleased to say that the drug was positive in all of those respects. So the patients found it was reasonably well tolerated, produced no significant side effects. It didn't interfere with the medication for Parkinson's disease at all. It got into the brain and it did the job that we wanted it to do, which was to reduce alpha-synuclein. So this trial was very positive, and that is what has led us now to the next study. Selen, if you could show the next slide, please. So that has brought us now to Ambroxol in what we've called the AsPro-PD study, which is Ambroxol to slow progression of Parkinson's disease. And this is a major first in the treatment of Parkinson's disease. This is the first trial to get to what we call phase 3 of a drug which is targeted specifically to a genetic related pathway of Parkinson's disease. So it's a hugely important trial based on a lot of work in the laboratory, but also our preliminary study. And we're going to launch this clinical trial next year. We're going to hopefully start recruiting people for the trial around May. So this study, AsPro-PD, which is really importantly supported by PD Frontline, the people who will be recruited into the Ambroxol study will come through PD Frontline. So it's what we call a phase 3 double-blind randomized placebo-controlled study. So it's a gold standard study of testing the effectiveness of a drug in clinical trial. The people that we're going to look for to recruit, of course, have to have Parkinson's disease, but we are going to take those with and those without a GBA mutation. Half of our Parkinson patients will have a mutation, the other half will not, which is, of course, is why we need to know the GBA gene result through PD Frontline. We're going to be giving the drug Ambroxol, and we'll obviously need to compare the effect of the drug with what we call a placebo, otherwise often referred to as a sugar type tablet. Basically to ensure that what we're seeing in terms of results are real. years is what we call the MDS UPDRS parts 1 to 3. So what will happen to people who are recruited into the study is that they will start Ambroxol or placebo and will continue on that for 2 years. During that time, we'll see them every 20 weeks. So not a particularly onerous schedule of visits. So only need to come and see whoever is at the center for the trial site every 20 weeks. At the end of 2 years, everyone will be converted to Ambroxol, and they'll know that they're on Ambroxol at that point and continue on that for a further 6 months, and we'll see them again at the end of that. Just an explanation of what we're looking at as this primary endpoint, parts 1, 2, and 3. This is a combination of the effects of the drug on quality of life, activities of daily living, and motor function. And we've talked importantly to the regulatory agencies to check what they will be looking for if they want to consider giving Ambroxol a license for Parkinson's disease. So we believe that we've designed the study as best we can to prove whether Ambroxol is positive or not. And of course, we don't know what the end result of the study will be. We'll obviously hope it's going to be positive, but the whole purpose of this trial is to see whether the Ambroxol really is capable of slowing down Parkinson's disease. People who come into the study will be able to continue their medication as normal. So we're not asking people to change their Parkinson's treatment at all. They'll just be added with Ambroxol or alternatively the placebo. And of course, they won't be aware which of those it is. So that, I think, is the next slide. Selen, if there's one more, there isn't. Perfect. That's the end of the story. Please let me know if you have any questions. If you're keen to consider the Ambroxol study, please go through Frontline. We do have a link on there which will help tell you a little bit more about the study. And early next year, we will be launching with Cure Parkinson's, who are the major sponsor of the study, although they are part of a sponsor consortium. But we will next year be launching more publicity about the trial to help inform everybody. Thank you very much.